Good morning, everyone. Thank you for joining us today for our latest webinar. The title is Identifying Translational Biomarkers for Diagnostics and Drug Development using Breath Biopsy and Vitro Headspace Analysis. I'm delighted to welcome my colleagues Huw, Agi, and Giuseppe, who will be running through some of the latest data we have here at Owlstone Medical. We'll make sure we have time for questions at the end. So if you do have questions as we go through the webinar, please submit that through the Q&A button at the bottom. And if you've got any technical problems, use the chat function to let one of the Owlstone team know. So please do feel free to post those questions as you go through, and we'll make sure that we have time at the end. Thanks, and enjoy the webinar.
So thank you very much for joining this webinar today on identifying translational biomarkers for diagnostics and drug development using breath biopsy in vitro headspace analysis. My name is Huw Davies. I'm heading up the sales team here at Owlstone. And I'm just going to give you a very short presentation on the company itself and the technologies we've developed for breath analysis before I hand on to my colleagues Agi and Giuseppe, who will run through case studies on this exciting new in vitro headspace capability. So Owlstone Medical is developing breath biopsy as a technology for the analysis of biomarkers on breath, really to support our customers through the provision of market-leading breath research products and services, but also to support our company's core mission to save 100,000 lives through the development of non-invasive diagnostic tests for early disease detection and precision medicine applications. So Owlstone Inc., parent company, was spun out from Cambridge University in 2004, really to focus on military and industrial applications, measuring volatile organic compounds, or VOCs, in the environmental air. Owlstone Medical was spun out in March of 2016 to develop the breath biopsy technology for measuring those VOCs on breath. So we have over 15 years of experience in VOC analysis across a range of different industries. And since Owlstone was founded, it's raised over 150 million in external investment, which has really allowed us to build up a very multidisciplinary team of 200 people headquartered in Cambridge in the UK. It's also allowed us to build out the world's first high-volume breath biopsy lab, and we're also running the world's largest breath-based clinical trials. So the technology is in use in over 100 clinics around the world for various research applications, and that's led to the production of more than 120 peer-reviewed publications that come from our own internal work as well as that of our customers. So Owlstone's focus is very much on the analysis of these small molecule volatile organic compounds. Human breath contains hundreds of these so-called VOCs that can originate from a variety of different sources. Importantly, many of these VOCs are end products of human metabolism or microbial metabolism, such as that found in the microbiome in the gut, and so can reflect patient phenotypes and lead to the discovery of novel non-invasive biomarkers. VOCs that we measure are not just coming from the lungs and the airways, they can also come from other organs and tissues in the body, carried by the blood through the alveoli and then into the airways and onto the breath for detection. So technology is not just limited to respiratory diseases, but can be applied to a wide range of other diseases affecting other organs in the body. So I spoke a little bit there about the endogenous VOCs that we can measure on breath, but of course they can also come from exogenous sources, which can include the air we breathe, the food we eat, the medications that we may be taking, as well as the microbiome, as I mentioned. So in total, we can measure all of these volatile organic compounds with our flat platform in one shot and compare how they change, for example, in different disease states. So some of the benefits of breath analysis, of course, it's completely non-invasive, so it's well tolerated and accepted by patients. It also has the potential to be collected anytime, any place, including at home, even with point of care type sensor devices that I'll talk about more in a moment. As I said, VOCs can originate anywhere in the body, really making them applicable to a wide range of early detection and precision medicine applications. So it's a very versatile technology for non-invasive biomarker research and ultimately diagnostics. So we developed workflows that allow us to measure these endogenous and exogenous chemicals, wide range over a large number of samples. But we're also taking more targeted approaches. For example, the evoke probe approach. So evoke stands for exogenous VOC. It's generally recognized as safe compound that we can give, for example, in a liquid or capsule form orally. That will then be metabolized by a specific enzyme, and then by measuring the substrate and product of that reaction, then gives us a non-invasive readout of a specific enzyme in a very sensitive and specific way. And that's an approach we're applying within our internal test programs in liver disease and early detection of lung cancer that Giuseppe will talk about more in his section. So we work with a wide range of different customers and partners. We work with a lot of the major academic clinical medical centers, both in Europe as well as in the US, as well as major pharma and biotech companies as well that are interested in detecting non-invasive biomarkers to support drug development. And we have capabilities within the business both to discover biomarkers, but also to deploy them. On the discovery side, we're taking principally top-down approaches where we can profile a large number of molecules very consistently, see how they relate or correlate with a particular phenotype or disease state, and thereby allow us to identify a potential biomarker. We're also, within our own test programs, taking more of a bottom-up approach where we can deploy the so-called evoke probes that I mentioned previously, but also looking at other pathway analysis type of approaches to elucidate volatile biomarkers that perhaps could be developed into evoke probes in the future, as well as the in vitro model work, which we'll talk about more in this webinar, which we're now opening up and providing as a service to pharmaceutical partners. Once biomarkers have been discovered and validated, the different routes for deployment, and we have a lot of those capabilities within Owlstone. For example, one option would be to develop a targeted mass spec-based assay, which could be offered through a central lab to a set of, say, 3 or 4 analytes of interest. We've gone through this process already. So one of the tests that we've set up as a commercial offering to the National Health Service and private healthcare is a food intolerance test where we're measuring hydrogen and methane on breath. So we've developed a kit which is sent out to the patient's home. They breathe into a tube. Hydrogen methane gases are collected. The tube is then sent back to our lab in Cambridge where we can do the mass spec analysis, and the output from that is then sent to our clinical partner at the functional gut clinic, and they will then be responsible for interpreting the data and providing feedback back to the patient. So that testing service is really set up for people with gastrointestinal conditions such as small intestinal bacterial overload, or SIBO as it's called, or carbohydrate malabsorption, to work with the patient to try and rectify those conditions. We also, for the future, are building out point of care devices that are sensor based, handheld, where the volatiles could be both collected and measured near patient or even a home environment. Of course, that does require us to discover and validate volatile organic chemical biomarkers, which then can be ported to that point of care device. And the type of sensor that would be used would be contingent on the type of volatile that we want to measure. But just to say that we have this sort of suite of capabilities within the business. Talking about our main discovery workflow, breath biopsy, Omni, these are the key components. This was really set up to address some of the challenges of the breath research field, have a standardized approach which allows for a very reproducible and reliable collection of a breath sample, and then the subsequent comprehensive analysis using mass spectrometry. So just to briefly talk through this, there's a breath biopsy collection station, which includes the receiver breath sampler, which allows for a reliable collection of a large volume of breath, pre-concentrating and stabilizing those volatile organic compounds collected from the patient onto a breath biopsy cartridge, which is effectively sorbent tubes, which can then be shipped back to our lab in Cambridge. Important to that whole setup is also the Casper portable air supply. So that is there to effectively filter out the VOCs that are present in the room where the sample is being collected, and it provides a filtered air supply into the receiver. So the patient is actually breathing filtered air, and that could reduce a lot of the background false positive and false negatives that can come from the environmental air, typically in a normal breath workflow. And that's all powered by a laptop and software It comes as part of that breath biopsy collection station package. The samples are then, once collected, shipped to our lab, ambient temperature on those sorbent tubes. When it gets to the lab, we can then perform high resolution thermal desorption GCMS analysis to give a very broad readout of all of the volatile organic compounds that are on that sample and start to measure fold changes that could be reflective of those VOCs being biomarkers for a particular disease. So this slide kind of talks through that in a little bit more detail. Samples arrive at our lab. We do the dry purging to drive off the moisture, inject internal standards. Those samples are then stored until we need to analyze them. They then run through the thermal desorption GCMS analysis, profiling somewhere in the region of about 500 or so on-breath compounds. We always include blank samples in every study to make sure that what we're measuring is actually physically on breath and not coming from some other source, whether that's the environment or the equipment we're using. It's absolutely critical. And that method's really then been optimized to maximize the number of on-breath VOCs that we can detect. We can then extract the data from the mass spec raw data, effectively normalize it, and then search against publicly available libraries such as NIST, which gives us a tentative assignment of the identification. We also are building a growing breath biopsy library of standards, which provides us a much more high confidence VOC assignment. So that's a resource that we're building in-house over time. Of course, we have the usual calibration standards and internal standards, so we can really monitor the quality control of the process and be confident in the data that's being produced. We also have a team of clinical and translational scientists that can advise our customers on optimal study design, making sure it's sufficiently powered, etc., making sure we have the right control groups in there alongside the disease groups that we're interested in. And then on the back end, they can perform the statistical analysis and biological interpretation and reporting back to the customer, providing those biological insights that come from the identification of the constituent VOCs and suggest biomarkers that could then be followed up in further studies. So that's our kind of human breath discovery workflow that we call Omni. More recently, we've developed this in vitro sampling capabilities that we're going to talk about more today. And Agi and Giuseppe will be going through some specific case studies around this. But effectively, our breath biopsy lab is equipped to do both the in vivo as well as this in vitro work using high resolution GCMS sample analysis. So in addition to that breath work, as I said, we can also analyze headspace from cell culture media samples. These can be collected in your lab, frozen, then shipped to us on dry ice, appropriate aliquots. We also have the capabilities in some cases to perform cell culture collection and headspace analysis ourselves in-house as well. So this in vitro analysis can support breath biopsy studies by identifying non-invasive biomarker candidates in vitro that, because they're volatile, have the potential to be translated to breath and put into human drug trials, whether they're non-interventional or interventional trials. This also provides insights to inform targeted analysis around a specific set of molecules that are elucidated from this type of analysis. So I think this is a really promising approach, which potentially allows the assessment of drug candidate pharmacology and the elucidation of volatile biomarkers that ultimately could be translated to breath. And as I said, Agi and Giuseppe will now be going through some specific case studies in this area.
Hello, my name is Giuseppe. I am a senior translational scientist at Owlstone Medical, and today I'm going to talk to you about Headspace assessment of potential EVOC probes to detect metabolic changes relevant to chronic liver diseases. As we know, there are several factors that can induce chronic liver diseases. One of these factors is excessive accumulation of fat in the liver, a condition known as non-alcoholic fatty liver disease. This is a benign condition that doesn't induce symptoms often. However, it can progress to non-alcoholic steatohepatitis, also known as NASH, which is characterized by inflammation and accumulation of fibrotic tissue. When the fibrotic tissue becomes excessive, then we talk about a condition called cirrhosis, which is the end-stage disease that can also lead to complications such as hepatocellular carcinoma, liver failure, and hepatic encephalopathy. The objective of Owlstone Medical is to develop a breath test that is able to differentiate subjects with NASH from subjects with non-alcoholic fatty liver disease. And there is a pressing need for a test at this stage of this disease because for now the only diagnostic method approved for NASH diagnosis is liver biopsy, which is an invasive procedure that can lead to complications. So we started comparing the breath of subjects with cirrhosis against subjects with healthy controls to increase chances to identify breath biomarkers that then can be back translated to earlier stages of liver diseases. So we began with the available data from the literature reporting that limonene is a potential breath biomarker found elevated in the breath of subjects with cirrhosis in different clinical trials. So the first step we took was to validate the results, and these are data that we published in 2020. And we also showed that limonene increases in the breath of subjects with cirrhosis, as we can see in panel A. And we also explored that limonene has also a diagnostic potential, as we can see in the rock plot built in panel B. We also learned that limonene correlates to the severity of the cirrhosis calculated as Child-Pugh class, as we can see in panel C. So, consistent with results shown in panel C, we found that limonene correlates with levels of bilirubin, INR, and albumin in the blood, indicating that limonene may be a biomarker that could be used as a readout of the functional capacity of the liver. Another additional important piece of information that we have learned is that the amount of limonene in the breath of subjects with cirrhosis depends not only on the severity of the disease, but also on how much the subjects are exposed to limonene. Now, this compound is abundant in orange juice, and we did a crude estimation asking the subjects how often they were drinking orange juice. And we found that those drinking it daily had higher levels of limonene in breath compared to those drinking it less frequently. This result suggests that we need to standardize exposure to exogenous compound to increase test performance and allow detection of earlier stages of liver disease. For this reason, we are now developing the EvoProbe approach, in which a compound, such as limonene, for example, is given to the subject, and then the compound itself or their bioproducts are measured in breath after administration. So the working hypothesis is that after an overnight fasting, we observe very low levels of these compounds in breath. However, after administration, we see a spike followed by a reduction. Now, given this reduction depends on how efficiently the liver is metabolizing these compounds, when in healthy subjects, we expect to see a faster reduction. However, when there is a liver disease, we expect to see a slower reduction. And this difference should allow to differentiate subjects with a liver disease from healthy subjects. This approach also has several advantages because we can use compounds with known safety profile, known as GRAS compounds, generally recognized as safe, and they are widely used in the food industry. We also can assess several metabolic pathways at once by using several different compounds. And also we will get a functional hepatic readout from the test. So to identify additional compounds besides limonene, we used a top-down approach in which we found several compounds that are upregulated in the breath of subjects with cirrhosis and some compounds that are downregulated, as we can see in this volcano plot showing on the x-axis the log 2-fold change, and on the y-axis the log 10 p-value. We also used the bottom-up approach, as we can see on the right, where we looked at the hepatic RNA expression datasets from different studies, And we found several genes that were differentially expressed in agreement between these different studies. And we could target some of those genes with specific compounds to detect their activity in vivo through the breath. Of course, because before performing clinical trials to assess these compounds, we established some in vitro approaches to identify the better compounds that can be used for the EvoProp approach, and also to screen potential diagnostic value between healthy and NASH models. And we are going to see some of these results. So in this example, we started from one extreme condition in which we have generated a knockout cell for one of the target enzymes that we found differentially expressed in NASH. We can see here in this Western blot that we have generated different clones that are knockout for this enzyme. We used 3 of these clones for metabolic screening of EVOX. So the experimental design is quite simple. So once we got the knockout, we seed them in 24-well plate. Then we add the VOC substrates we are interested in. Then we collect aliquots at different time points, and from these aliquots we can measure in the Headspace how much of the substrate has been converted to the expected bioproduct. What I would like to highlight as well is that all the cell culture part has been performed elsewhere off-site, not at Owlstone Medical. And at Owlstone Medical we have performed only the Headspace measurement. And this is very convenient for us because we can assess cell culture systems that have been already established elsewhere without the need to import them in-house and establish the capability for these cell culture systems here at Owlstone Medical. So to show some results, we can see here that for this compound called substrate 1, we can see that we observe a reduction over time, which is expressed on the x-axis, and the amount of the substrate expressed on the y-axis. So we can see that when we have only media, we observe some evaporation, which is expected because this is a volatile compound. However, when we have cells, either if they are wild type or knockout for the target enzyme, we can see that the substrate reduces over time. And that's also expected because this compound is known to be metabolized by several metabolic pathways, not only the enzyme that we have depleted from these cells. However, if we look at the specific bioproduct, we can see that in the absence of cells, so when there is only media, there is basically no production of the bioproduct. In wild-type cells, we see a spike of the bioproduct half an hour after the treatment. And in the knockout cell lines, we can see that the production of the bioproduct is reduced. However, we still see some bioproduct, and that's due to probably to other isoforms of this enzyme that are mediating a similar reaction. However, we also investigated additional bioproducts to identify some that may have a higher affinity for our target enzyme and therefore provide better results. And I'm going to show you an example here. So again, for this other compound called substrate 2, we can see similar results as before. So a reduction over time in the media and a stronger reduction when there are wild type and knockout cells. However, if we look at the specific bioproduct, in this case, we can see in the wild-type cells, spike of the bioproduct after half an hour. And then in the knockout cell lines, we can see that the production of the bioproduct is reduced, indicating that this compound may be more specific for this enzyme and therefore may be a better reporter in vivo for the detection of the activity of this enzyme. Of course, comparing wild-type and knockout cells is quite an extreme condition. And therefore, the next question we asked is if we could see some drug modulation for the enzyme activity using a Headspace analysis. For this purpose, we used a drug inhibitor for our target enzyme. And then we have, as before, we have added the VOC substrate we are investigating, collected the different time points and measured that space. And again, also in this case, all the cell culture part has been conducted off-site. We took advantage of systems and models that were already established elsewhere. So, to show some results, we can see here that again, for this called substrate 2, we see a reduction over time due to evaporation, either if there is the media or if there are also the cells. However, we can see when we look at the bioproduct, we can see that where there is the media and where is the drug inhibitor, the bioproduct is basically absent. And where we add the vacuum in which the drug was dissolved, we can see that we observed a spike of the bioproduct over time. The amount of drug inhibitor used here was estimated to be an IC75, and we are now in the process of conducting experiments with lower concentration of drug to evaluate a dose response. Still, we were also looking to see if we could differentiate more subtle differences, for example, those that may be generated by a disease, such as, for example, non-alcoholic steatohepatitis. To address this question, we used the CNBio model system, organ on a chip, in which human hepatocytes are cultured on a scaffold where they can polarize and stay differentiated up to 3 weeks. Then the media flows through the hepatocytes and to provide oxygen and nutrients. So the advantage of this model is that we can not only have healthy hepatocytes, but with specific treatments, we can generate also models of different stages of non-alcoholic fatty liver disease. For example, if we add only fat, then the hepatocytes accumulate fat, and they could represent non-alcoholic fatty liver disease at the stage of simple steatosis. If we add also Kupffer cells and hepatic stellate cells, then we would have accumulation of fibrotic tissue mimicking a condition such as NASH. And then if we add also TGF-beta, then we can generate a condition that resembles a more advanced stage of NASH, almost cirrhosis. So we started by comparing NASH hepatocytes against healthy hepatocytes. So the experimental setup was as follows. So we, once the hepatocytes were differentiated, cultured, and they were expressing a healthy phenotype or a NASH phenotype, we added compounds we were interested in to the culture media, and then collected it at different time points to measure the substrates and bioproducts. We can see here levels of TIMP1 and interleukin-6 in the supernatants as a readout of the establishment of the NASH phenotype, because TIMP1 is a marker of fibrosis and interleukin-6 is a marker of inflammation. So if we look at one of the substrates that we have been using, so we can see that we observed a reduction over time, also in the absence of hepatocytes, because this is a volatile compound that evaporates over time. However, when there are cells, we see a stronger reduction. If we look at the bioproduct for this substrate, we can see that in the absence of hepatocytes, we have no production of the bioproduct, which is the flat line, the gray flat line at the bottom. Then when we have healthy hepatocytes, we observed a spike of the bioproduct 6 hours after the treatment, followed by a reduction, which is probably due to secondary metabolism. However, when we have NASH hepatocytes, we see that the production is slower, And also the secondary metabolism is lower, showing a peak after 24 hours. So in conclusion, we showed different applications for the in vitro VOCs. And of course, this approach has has a lot of potential, for example, for preclinical assessment of target engagement, functional phenotyping following genetic perturbations, functional metabolic characterization after drug treatment. And of course, I would like to highlight that this approach, so the VOCs that are identified in preclinical work, have the potential for translation to clinical studies. That's because the compounds that we tested are all GRAS compounds, as I mentioned before, and they can be given to people, and we can measure a spike in breath after administration. For example, as we can see here for these compounds, where we see very low levels after an overnight fasting, and then in healthy subjects, after administration, we see a spike in breath followed by reduction. And by using Omni, we also identified a potential bioproduct for these substrates that could be generated by the metabolism of these compounds. Thank you very much for your attention. And I would like to acknowledge all colleagues that contributed to generate this data.
Welcome, everyone. It is my honor to present you with our latest work on in vitro headspace analysis to investigate the inflammation response in bronchial cells. As we all know, there are different types of lung diseases, and looking at how many people worldwide are affected by those different lung diseases, how many people are each year diagnosed, how many people die because of those lung diseases, and of course, how much money it costs to society, it is pretty understandable that lung diseases are the topic of various studies on academical level, but also on industrial level. The way how we can investigate lung diseases are, of course, different. We can do it using in vitro models, we can do animal models, and of course, human models. And if we look at the in vitro models, there are also different ways of doing it. And I would like to show you today in vitro model called MUSIL-AIR, which is in vitro 3D model, is an air-liquid interface. It's a cell model of the human airway epithelia, and it's a 3D epithelium of the human upper respiratory tract made of primary low passage cells cultured at the air liquid interface. As you can see on the figure here, the in vitro 3D model has 3 cell types. So we have a basal cells, we have a goblet cells, and we have silated cells. And as you can see here, the top layer is basically covered with mucus. The system here consists of about 500,000 cells. So if we compare it to, of course, very traditional flask cultures, which have like 30 million, we can say that this system is pretty limited. This in vitro 3D model can be used with 12, 24, or 96 well plates. We used 24 well plates, and as indicated before, at Owlstone we have different types of headspace approaches, using basically more manual approach, HiSorb, where we have the probes that absorb the volatile compounds from the headspace, and more automated Sentry, where we have again the same probes that absorb the fogs that collects, that are basically in the headspace of the samples. What is pretty important to notice here, we really work with a limited volume of the sample here because only 50 microliters. But just to maybe further indicate, we work here with the liquid, but of course the system can be also used for solid samples here as well. So before we move towards the 3D in vitro model, we first wanted to check our analytical variation analytical reproducibility of the system. So we started with simple samples. So we used 3 compounds, 50 microliter of samples, 3 compounds in PBS, and we sampled for 30 minutes at 38 degrees, and then we look at our results. And this is what you can see on the figure on right, where you see the areas of those 3 compounds. An area here can be considered as a month of the compound in measured samples. And as you can see here, there is really not that much variation between our replicates. And if we change it, show it as a coefficient of variation, we see that there is pretty good analytical variation of about 5%. This is, of course, pretty simple samples, so only 3 compounds. So therefore, we moved further, and we used a mixture of 23 compounds. We use exactly the same methodology, so half an hour sampling at 38 degrees, 50 microliters of sample. And you can see the results in the table on the right with different compounds and the variation that they showed. And 21 out of 23 compounds did pass our quality check. There were 2 compounds that did not pass our quality control because they had pretty high coefficient of variation. And overall, from those 23 compounds, we had a median coefficient of variation of 6%. So having the analytical method established and checked, we were feeling comfortable to move towards basically in vitro model, where we used the Musil R in vitro 3D model to look at inflammation. And we induce inflammation in bronchial cells using bleomycin and LPS. And just to indicate that bleomycin is an anti-cancer drug that triggers inflammation and promotes ROS formation, and LPS is a bacterial soluble component that triggers innate immunity response. So what did we exactly do here? So we had our air-liquid interface in vitro assay. We basically use bleomycin and LPS that was added to the media of the cells, and we sampled the media and mucus. So just again to say that the mucus is basically what is covered with the top layer of the cells, and we collected the headspace from media and from the mucus samples. To give you a bit more details here, we in our 3D in vitro model, we use healthy bronchial cells, as can be seen in the table on the figure here. So we collected mucus and collected medium. We had different treatment group. As I said before, we used LPS and bleomycin that were used to induce inflammation, but we also needed some control samples. So we had cells only and cells treated with PBS. And here PBS, because PBS was used as a vehicle here. As you see on the sampling days, so we use 2 sampling time points. We had day 0, so it's a baseline, so it's before the cells were treated. And then we had after 48 hours, so day 2 of after treating the cells with bleomycin and LPS. So let's start from looking at the results obtained for the media samples. So we collected the media at day 2 time points. We measured the headspace, we collected the headspace, we measured the samples, and we were able to detect 391 different volatile organic compounds that were excreted in the media samples. We analyzed the data, we check if we see some natural groupings in the data and if we see some statistically significant differences. And this is what you can see here on the figure on the PCA score plot. where each point here is basically a single sample. And I think the first conclusion that can be made from this figure is that the samples really cluster with respect to the treatment group. So we have our LPS samples, we have our bleomycin, and here we have the control samples, and the control samples are the samples with cells only and cells treated with PBS. We can make this figure looking maybe a bit more clear and understandable, and I can basically take each of this point and project on my axis and represent this figure as a box plot. And this here, I think you can clearly see that my control samples are similar, while I see the differences in the cells treated with LPS and the cells treated with bleomycin. So when we look at which compounds specifically cause those differences, we saw some alkanes being elevated in cells treated with LPS. And then we saw some, for instance, some aldehydes that were related more to bleomycin. So what we notice here that there are some volatile organic compounds that move differentially between LPS and bleomycin treated cells. And that may suggest that maybe there is a bit of a different underlying pathway engaged by LPS and bleomycin. And of course, we did not go in depth into structural elucidation of the compounds that were responsible for the groupings, but we know that those were alkanes and aldehydes, and we know that those are the markers of inflammation. So from the biological point of view, that of course makes sense, because we know that LPS and bleomycin are supposed to induce inflammation here. So those are the results for the media samples. As I indicated, we also sample mucus samples. So here, the samples from the mucus, we detected a bit less volatile organic compounds that we did with media, because only 336 different compounds were detected. We here look at the differences observed at between day 0 and day 2 samples, also between the different treatment. And as you can see on the figure here, so this is again the PCE score plot, where each point here is a single sample. And again, we can clearly see 3 clusters. So we see our bleomycin, we see our LPS, and we see our control samples. And again, control samples are PBS samples. And again, to represent it a bit in more easy way, we can put it as a box plot. So we see that the controls are similar, while cells treated with LPS or bleomycin are basically different. So we had here also the samples of day 0. And what we did, basically, we took those samples and we projected in the previous PCA model. And of course, if all goes well, the samples of day 0 should cluster with our control, because there shouldn't be much happening, right? Those are the control samples as well. And that's what you can see basically here, because the cells at day 0, which are marked with the black field cycles, they do project into our controls. And again, if I would like to represent it as a box plot, you see that the controls are looking the same as the cells and cells with PBS at day 2. So to look at specific compounds that are responsible for those differences, we see some compounds that are specific for LPS and bleomycin. So we see here alkane for LPS and octanol for bleomycin, but we have also some compounds that basically are elevated in cells after treating with bleomycin or LPS. To basically a bit summarize of what I told you so far, so we see a distinct changes in the VOX profile of cells treated with bleomycin and LPS after 48 hours, we see some compounds that are specific for LPS or bleomycin, but there are some compounds that basically are different for both treatments. What is pretty important to say here that if we compare the results obtained from mucus and media, we notice that for the mucus samples, variation between replicates were much, much higher. And that's pretty understandable because the amount of mucus that is collected above the cells is really limited. So this is probably the cause of the variation. The compounds that we saw changing due to inflammation are mostly aldehydes and alkanes. Of course, there has to be a further work to verify the specific identities of those folks. In the previous experiment that I just have shown you, we used just one time point, right? So it was pretty simple setup. We had baseline samples, then we treated our samples, and after 2 days we checked, like, okay, what happened after inducing inflammation for 2 days. However, the 2 days is pretty long time after stimulating the samples. So it can be that if we look at the earlier time point, we might see maybe larger effect size. So therefore, we decided to look if we can create kind of a 3D in vitro model where we could sample over time, or we could sample more frequently the headspace. So that's what we did. We had our cells, and we use here only bleomycin and PBS as our control. And we basically had to a bit change our sampling approach, but we basically modified our headspace sampling where we could use the molecular cells inside the sampling vessels to be able to basically sample over time. And this is what we basically did. We sampled after 1 hour, 4 hours, and 24 hours of treatment. And here you can see the results. So you see the PCA score plot again, and each of the points here is a single sample. And what you see here is that our samples with PBS and bleomycin, they basically are very well separated after 1 hour. Here we have samples after 4 hours and 24 hours. And if we look at the further PCAs, we see the differences between samples after 24 hours. So basically what those two figures tell us is that we see larger differences between PBS and bleomycin after 1 hour, and we saw the increase in aldehydes levels. We also see the differences at 4 hours and 24 hours, but a bit with smaller differences here. And to summarize, we were able to create a bit of custom method to sample Fox headspace over time. And just to emphasize here, that was done directly from the culture cells. We can, of course, collect at different time points. We look here at the 3 time points, but it is possible to basically do more time points here. We saw here very large differences at the early time points in comparison to the later. And of course, all the results that I showed now, there has to be a further work to see how, for instance, this could be performed on the living cell. But if we would like to, for instance, sample much longer, for instance, maybe for 3-4 days, and then of course that the cells still stay alive. And with that, I would like to thank you for your attention, and I will be happy to answer any questions.
So the first question that's come through is, so it sounds like models can be established elsewhere. Can we send the samples to you? What types of samples and how long does it take to process? So maybe Huw, you could start us with that.
Yes, well, with the in vitro samples, then yes, samples can be processed in your lab using whatever cell culture system you have established there. So it's fairly flexible in that regard. Typically, what we would do is have you would collect the media samples from those cells, so absent from the cells themselves, and then aliquot it out and ship it to us on dry ice. When it's received at our lab, then we would do the Headspace analysis once we've defrosted those samples. So that's typically the way it works. With the breath analysis side of things, we actually would place a breath biopsy collection station in your lab, and you would perform the breath collection following training from our side, and then the samples would be shipped back to us in the breath biopsy cartridge, as I explained in my introduction.
Excellent, thank you. Next question for Giuseppe. Diabetes and obesity are strongly associated with the etiology of NASH. Can you discuss how these metabolic states may affect your model?
Yeah, so basically the NASH phenotype that CN Bio is able to reproduce in vitro at the transcriptional level overlaps with the gene expression that has been measured in humans. So it's quite a well-recognized NASH phenotype. Of course, this is a very interesting question, and probably we should get back to them and ask if they have done specific experiments to characterize the insulin resistance when they induce a non-alcoholic fatty liver disease of a NASH phenotype. What I am expecting is that the response to insulin will be impaired in these ill hepatocytes compared to healthy hepatocytes.
Great. Next question, I think one for Agi. Bleomycin represents a very severe model to induce inflammatory lung processes. Is this approach translatable to humans?
Thanks for the question. Yes, of course, you are right. Bleomycin is pretty, intense type of the model. Just to mention here, we did not use such a high concentration of bleomycin. So in this sense, yes, it is transferable to humans. And also, I mean, you can look at this inflammation on different levels, right? So as we show the case where we look over time, then also different, let's say, level of inflammation, and then also fibrosis would be here developed. So yes, definitely it is translatable to humans.
Another related question, I think, for you, Agi, as well. In your time-resolved experiment, do you purge the cells with oxygen?
So in this experiment, no, we did not. What we did use was basically equilibrated media, and because we really work with very limited number of cells, because that was just 500,000, that was basically enough to keep the cells alive. But of course, if we would go to the larger system, then yes, it would have to be flushed to keep the cells happy.
Brilliant. How do you find target exogenous probes for specific applications? I think, Giuseppe?
Yeah, so in our case, we use different approaches. So we started from looking what was available there and was from other studies. And then we also used the breath biopsy only with an untargeted approach to identify compounds that then could be translatable for the EVOC approach. And besides that, we also looked into transcriptomics data to identify enzymes that metabolize compounds that could be used for the EVOC approach. So yeah, from this point of view, we just need to get creative and identify compounds that are suitable for Headspace analysis and translatable to human in clinical stages.
Great. Couple of technical questions questions. So one is around the type of column and mass spec system used, and what type of dynamic range do we get on that system? Maybe Agi, start on that, please.
Yeah, so in respect to the column used, so we often use Stabilvax column here. With the dynamic range, I mean, we work with the Orbitrap system here. So yeah, if we talk with like TOF, which has like around 1,000 dynamic range, here we talk about 5,000.
Great. Anything to add, Giuseppe, on those? No. Okay. One question probably for you on the NAFL-NICE model. It's interesting. I missed how you were able to measure IL-6 and TIMP. What is the source of the cytokine?
Yeah, so to generate the NASH phenotype, hepatocytes are co-cultured with the hepatic stellate cells, which are responsible for the fibrotic tissue formation, and then with the Kupffer cells that are responsible for the inflammatory response. And then aliquots of media are collected, and the biomarkers are measured using routine kits.
Great. There's a question about our approach to normalize the data. Giuseppe, do you want to take that one as well?
Yeah, sure. So in terms of normalization, we focus mainly on taking into account the batch effect. So making sure that we balance the sequences that we run, and so in the way that we exclude the variations that is due to the batch effects. In terms of intra and inter-subjects variability, so for now, we basically, we don't normalize for that at the moment because we are looking for biomarkers that have an effect size which is higher than the intra and inter-subjects variability, so that so those would be like more robust biomarkers overcoming these confounders.
What's the advantage of considering VOC biomarkers to other classes of biomarker? Huw, maybe you could take that one for us, please.
Yeah, I mean, I think there's lots of different ways to answer that question. Of course, volatiles on breath and the way we measure it is completely pain-free and well tolerated, obviously non-invasive, so all the benefits that come from that. I think the other thing I would add is that because we're identifying what they are, obviously we can relate it to the biology. And I think we can view this as a data set that's perhaps completely orthogonal to other types of analysis, whether that's genomics, metabolomics, or proteomics. I think it adds value in that regard as well.
Great. If we want to run or discuss an in vitro or in human study, what are the next steps? Huw, again on that one, please.
Yeah, everybody that's attended this webinar today will be getting an email thanking them for their involvement and also providing a recording of the webinar. And so you could respond directly to that email, or you could also send an email to breathbiopsy@owlstone.co.uk as well. And we'd be happy to talk to you about your study. We have a team of clinical and translational scientists here who can talk through specifics of your study and advise on the study design, and we can take it from there.
Excellent. So we've got about 30 seconds left. So just want to say a massive thank you to our speakers and for you all for attending today. Any questions we didn't get time to cover there, we will provide typed responses to those that will sit underneath the recorded webinar. As she said, we'll send out a link to the recorded webinar as well. So thanks again for your time and have a great rest of your day.